Alright guys, welcome to your third video of this unit. This video is going to talk about a process called transcription. So before we get into that, let's just recap. DNA is the genetic instructions that build organisms. What we mean by that is that DNA holds the instructions to code for proteins. I know that doesn't make a lot of sense right now. That's what we're going to get all into, but your body is made up of proteins, and DNA holds the instructions to build all of those different proteins. El DNA contiene las instrucciones para la construcción de proteins. As we said in the first video of Unit 3, DNA is stored in the nucleus. We know we have a cell, and inside a cell we have the nucleus. Inside of the nucleus, that is where DNA is, which we know is stored in chromosomes. So yeah, the nucleus is inside of a cell. Inside the nucleus, there are chromosomes, and chromosomes are made out of DNA. That's where DNA is held. So like we're saying, DNA is used to build proteins. That is a process called the central dogma. So the central dogma, this process where DNA builds proteins, it has two steps. The first step is called transcription, and the second step is called translation. Like we said, this video is about transcription. So we're going to zoom in on this first step of this overall process of how DNA builds proteins. So transcription happens for a couple reasons. Like we keep saying, DNA is stored in the nucleus and it cannot leave. DNA always, always, always stays in the nucleus of a cell. Now, this is a problem because the organelle that builds proteins is the ribosome, and that is outside of the nucleus. So imagine that you have all the instructions to build a puzzle or let's say build a Lego set or something like that, but you're stuck in one room and the Lego set is outside of that room. You would have to read the instructions, tell someone who can leave the room, and they can go build the Lego set outside. That is the same situation here. The DNA has the instructions, but the thing that they need to build is outside, not where the DNA is. The DNA is in the nucleus and it cannot leave. So the DNA has to send a messenger to tell the instructions to the ribosome. That messenger is called mRNA. That little M, it stands for messenger. So this process of transcription is when mRNA makes a copy or transcribes the instructions of DNA, which is in the form of these base pairs. So you can see here, DNA and RNA, they have different bases that they work on. So the DNA down here, the blue, has G, C, A, and T. When the RNA is transcribing the DNA, it matches up the matching bases or the corresponding bases with the DNA. So you guys remember from before, G goes with C, C goes with G, A usually goes with T, but RNA doesn't have T as one of its bases. It uses U instead. Everything else is the same, but RNA has U instead of T, no T on RNA. So when there's an A base on the DNA, the RNA puts a U there instead. So you can see it keeps building like this. Wherever there's a C on the DNA, the RNA gets a G. Wherever there's an A, the RNA gets a U. Look again, wherever there's a T on the DNA, the RNA gets an A. Whenever there's a G on the DNA, the RNA gets a C. That mRNA, that messenger RNA, then travels outside of the nucleus. You can see this wall, that's the nucleus wall. The RNA travels outside the nucleus down to the ribosome. this is where proteins are made. So just to say it again on another picture here, we have the DNA that stays up in the nucleus always, and then transcription occurs where RNA creates a copy of the DNA's message. Then that RNA that we just said, that mRNA, messenger RNA, can travel outside of the nucleus, and then it's going to go to the ribosome. All right, so key points here. Number 1, DNA cannot leave the nucleus, but it needs to get its message to the ribosome. So in order to communicate with the ribosome, DNA transcribes its message into instructions for RNA. Just to say that another way, to communicate the instructions to the ribosome, DNA transcribes its base pairs into mRNA. Para comunicar las instrucciones al ribosome, el DNA transcribe sus pares de bases en mRNA. Then the mRNA travels from the nucleus to the ribosome where it is used to build proteins. This step we will talk about in the next video, but right now we're just focused on transcription, the step where DNA sends mRNA as a messenger to the ribosome. And finally, DNA holds instructions for building proteins. The process of building proteins is called the central dogma, and it has 2 steps: transcription, where DNA transcribes its message into RNA, and translation, where mRNA is building proteins. One more thing I want to reiterate is that when RNA is transcribing DNA's message, it creates a strand with the corresponding base pairs. For example, if DNA has a G, RNA will put a C. If DNA has a C, RNA will put a G. However, when there's an A, RNA cannot put a T. RNA does not have T as a base pair. So when there is an A, RNA puts a U. So how would this DNA set of bases be transcribed into RNA? Where there was a G, we have a C. Where there was an A, we have a U. Where there was a T, we have an A. Where there was a C, we have a G. Where there was a G, we have a C. And last, where there was an A, we have a U. So if you guys have any questions about this, we of course know this RNA would then travel outside of the nucleus to the ribosome. And yeah, that's the whole process. I know that's a lot of information, but if you guys have any questions, please come to office hours or ask us in class. And rewatch this video. There's a lot of new vocab being thrown in this unit, so make sure you're staying on top of it. Remember, you guys need to know this stuff for class.